Good morning. It's April 24th and this is your daily brief in science. Here's everything you need to know. Recent research from the University of Michigan has unveiled new insights into corn genetics that could enhance crop productivity and resilience in changing environments. Published in the journal Science on April 18, 2025, this study marks a significant advancement in understanding plant genetics at the cellular level. Led by Alexandra Morand, the study analyzed DNA from nearly 200 maize lines, focusing on gene activity across different cell types. This analysis aimed to link molecular biology to observable traits such as ear quantity and size. The findings indicate that most phenotypic variation in maize arises from changes in gene regulation, how, when, and where genes are expressed, rather than from alterations in the genes themselves. Moran points out that previous studies struggled to connect genetic changes to observable traits, highlighting a critical gap in understanding plant genetics. He compares the findings to understanding a car's components and their interconnections, which aids in optimizing plant traits. Additionally, the study provides insights into the evolutionary changes in maize varieties, illustrating how regulatory sequence changes have influenced performance in diverse environments. This research also aids scientists in predicting the effects of genetic changes, allowing for strategic improvements to corn adaptability. The collaborative effort included researchers from the University of Georgia and the University of Munich, supported by funding from the National Institutes of Health and the National Science Foundation. Ultimately, the research underscores the importance of cellular context in gene usage, revealing that different cell types utilize the same genes in varied ways, affecting traits such as ear count and size in corn plants. Researchers have unveiled a groundbreaking catalyst structure that could revolutionize hydrogen production through water electrolysis. This new design features mesoporous single-crystalline cobalt-3 oxide, which is doped with atomically dispersed iridium, or IR. Led by Professor Hao Li, the study emphasizes the importance of the mesoporous architecture, which allows for effective single-atom IR loading and ensures stable catalytic activity. The innovative catalyst can achieve a high IR loading of 13.8 weight% without forming large IR clusters, a critical factor for enhancing performance. Its configuration promotes the formation of cobalt-1R bridge sites, which demonstrate high intrinsic activity under acidic oxygen evolution reaction conditions. Remarkably, this catalyst maintained its performance for over 100 hours, with an overpotential of just 248 millivolts, showcasing both stability and efficiency. The research also indicated a significant reduction in the leaching of IR and cobalt during reactions, with losses about 1/4 and 1/5 of those seen in traditional IR cobalt-3 oxide catalysts. Computational analysis revealed that oxygen intermediates cover the cobalt-3 oxide surfaces during reactions, passivating cobalt sites. However, the introduction of IR doping reactivates these sites and enhances the structural integrity of the catalyst. While iridium is known for its exceptional performance in the oxygen evolution reaction, its scarcity and high cost pose challenges for the scalability of electrolyzer technologies. Future research will aim to optimize the doping level, scale up the synthesis process, and integrate this catalyst into commercial electrolyzer systems. The findings, which merge experimental data with computational modeling, are available through the digital catalysis platform created by the Hao Li Lab. This study, published in the Journal of the American Chemical Society, presents a catalyst design that maximizes atomic-level efficiency and stability for hydrogen production. Lyme disease, caused by Borrelia bacteria transmitted through deer tick bites, affects nearly 500,000 people in the U.S. annually. If not treated early, it can lead to serious chronic health issues. As climate change continues to increase tick populations, the prevalence of Lyme disease highlights the urgent need for more effective treatments. Currently, the standard treatment for Lyme disease involves high doses of doxycycline, which disrupts the gut microbiome and does not selectively target Borrelia species. However, recent research from Northwestern University has identified piperacillin, an antibiotic commonly used for pneumonia, as a promising alternative. Piperacillin can effectively treat Lyme disease at a dose 100 times lower than doxycycline and has minimal impact on gut microbiota. In studies, Both piperacillin and doxycycline cleared the infection in mice, but piperacillin showed negligible effects on gut bacteria diversity compared to doxycycline. The research involved screening nearly 500 drugs to find those that specifically target Borrelia without harming other bacteria, leading to the selection of piperacillin as the most effective option. Brandon L. Jutras, who led the research, emphasizes the potential for personalized medicine in Lyme treatment, aiming for customized drugs based on specific strains of Borrelia. Despite the absence of an approved human vaccine for Lyme disease, ongoing research is focused on developing proactive strategies for diagnosis and treatment. While the results in mice are promising, researchers caution that antibiotic responses may differ in humans, with human trials for piperacillin planned for the coming years. Historically, piperacillin is used in combination with tazobactam for treating strep infections, but it works effectively alone against Lyme due to the unique characteristics of Borrelia. A recent study published in Nature Cell Biology sheds light on the aggressive nature of medulloblastoma, the most common malignant brain tumor in children. This research reveals how these tumors spread and grow on the leptomeninges, the membranes that encase the brain and spinal cord. Researchers from Baylor College of Medicine, Texas Children's Hospital, and the Hospital for Sick Children in Toronto have uncovered a communication pathway between metastatic medulloblastoma cells and leptomeningeal fibroblasts, which is crucial for tumor growth. The study shows that reprogrammed meningeal fibroblasts, unlike normal fibroblasts, secrete proteins BMP4 and BMP7 that promote tumor colonization and spread. Furthermore, metastatic medulloblastoma cells produce a protein called PDGF, which recruits and transforms leptomeningeal fibroblasts into tumor-specific fibroblasts that aid in tumor proliferation. Notably, blocking the PDGF signaling pathway with a neutralizing antibody significantly improved survival rates in animal models, indicating a potential therapeutic strategy. The findings suggest that interrupting the communication between tumor cells and fibroblasts may prevent disease progression. Importantly, the implications of this research could extend to other cancers, such as melanoma, breast, and lung cancers, that also metastasize to the leptomeninges. This study enhances our understanding of how tumor cells interact with surrounding cells, which could lead to new treatment avenues for medulloblastoma. Dr. Michael D. Taylor, the corresponding author, emphasized the significance of this discovery in revealing how tumor and surrounding cells collaborate to foster a supportive environment for disease progression. Co-first author Dr. Namal Abasundara pointed out that understanding these interactions is vital for developing improved treatments for children affected by this aggressive cancer, as metastases remain the leading cause of illness and death in these patients. Sodium-dehydroacetate, commonly known as NADHA, is widely used as a food preservative for its ability to inhibit microbial growth. However, recent studies have raised significant concerns regarding its potential health risks, including cancer, liver injury, and genotoxicity. Predictions from ADMET Lab 3.0 indicate that NADHA poses risks for genotoxicity, hepatotoxicity, and carcinogenicity, with inflammation and oxidative stress identified as central mechanisms contributing to its toxicity. Prior research has linked NADHA to various adverse health effects, such as weight loss, allergic dermatitis, and cardiovascular risks. This underscores the necessity for thorough safety evaluations of food additives. The multi-organ toxicity profile of NADHA challenges traditional safety assessments, highlighting the need for comprehensive evaluations to better understand the risks associated with food additives. Network analyses have identified 6 core targets related to NADHA-induced cancer and liver injury, as well as 4 core targets associated with genotoxicity. This suggests that its toxicity may impact multiple organs through shared pathways. Research utilizing network toxicology has pinpointed key molecular targets, including ALOX5, PTGER4, PTGS2, and SMAD3, which are linked to inflammation and oxidative stress. Gene ontology and KEGG pathway enrichment analyses reveal that NADHA activates harmful pathways, such as the AGER-AGE signaling pathway, which is associated with tissue damage and cancer development. Given these findings, the importance of experimental validation of NADHA's effects is emphasized to refine regulatory thresholds and ensure food safety. As a result of these safety concerns, the usage of NADHA in various food products will be banned starting February 8, 2025, following several incidents of contamination and health risks. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.